This is Unit 8, Molecular Genetics 8.3, Protein Synthesis. Let's get into this. Okay, protein synthesis. What is that? We are making proteins. That's it. Protein synthesis is composed of two separate processes: transcription, which happens in the nucleus, translation, which happens on the ribosome. Transcription is the production of a messenger RNA from the DNA. Translation is the production of a protein or a polypeptide using that messenger RNA, right? And this happens out in the cytoplasm on the ribosome, or it could happen on the rough ER, like a part of that rough endoplasmic reticulum, right? And so these two processes, very important, understanding the difference between them: transcription, making a messenger RNA; translation, taking that messenger RNA and then making a protein out of it. The whole thing then is called protein synthesis. Purpose of protein synthesis is to make proteins. Here they are, those two reactions. This is another helpful picture. Transcription, start with the DNA, end up with a messenger RNA. Translation, start with a messenger RNA, end up with a polypeptide. Think about it this way. If I ask you to open up a textbook in page 223 in the bottom right-hand corner, there's a paragraph, and I ask you to transcribe that paragraph, you immediately know what I'm asking you to do, right? You're making a copy of it. However, in the textbook, it's in like printed form. It's in a nice font, right? But I'm asking you to handwrite it. Same language, just different format, right? And so think about DNA to RNA as being same language, the language being nucleic acid, just a different format, going from this permanent copy of the DNA to this more just kind of a temporary copy of that gene being a single-stranded RNA. Right, and so you'll notice only one of those RNA or DNA strands are coded from, and we'll talk about that later. And then translation. What does translation mean? Going to a different language, right? And so I hand you that transcription that someone's copied, and I said, please translate that into, I don't know, Japanese. And then so someone who speaks Japanese is going to take that paragraph and is going to translate it then into another language. And so in this case, translation is taking the messenger RNA and translating it into the language of protein or polypeptide, which are made out of amino acids. That's what that met and illy and ser stand for down there. All right, so transcription, translations, two processes of protein synthesis. And remember, these are separate from DNA replication, which is replicating the DNA, making a copy of the DNA. Don't make a big amalgamation, repli, strip, translation. Don't do that. Three separate processes. Moving on, what is transcription? Transcription produces a complementary strand of RNA from the DNA. How does it do that? Well, there's this enzyme. It's called RNA polymerase. Sounds a lot like DNA polymerase, except what does DNA polymerase make? DNA. What does the RNA polymerase make? RNA. It's not hard. Don't make it hard. And so every time that RNA polymerase sees an A, what is it going to get? Oh, it's going to go get a U, right? Because there aren't any T's in RNA. C and G, A, or G and C, G, C, C, G, same. Every time it sees an A, it's going to get a U, but every time it sees a T, it's going to go get an A, right? Don't say a U, right? But so you'll need to be able to make a complementary strand of RNA from a piece of DNA. It's going to be a skill that I'm going to ask you to, could be a test question, is a test question, definitely 100% chance is a test question. And so here's your piece of RNA. It's being made slowly, one bit at a time. And then this is going to come to a place where it says stop, and then it's going to stop, and you're going to have this piece of RNA, right? And then that piece of RNA is then going to leave the nucleus. Once it leaves the nucleus, it's going to go find a ribosome and begin this process called translation, which happens on the ribosome. Purpose of the translation, again, is to make a string of amino acids, which is basically a protein. But there's some other things that we need to talk about, because notice this is just more complicated than transcription. So let's talk about some stuff. Well, first of all, you'll notice that it's being read in like little groups of three. Those little groups of three have a name. They're called codons. And so these groups of three pieces of messenger RNA, stand for a particular amino acid. So in this case, UUC stands for this PHE, or phenylalanine, which is this particular amino acid. These next three are going to stand for something else. The next three, something else, right? And you're just going to keep kind of going down the line until you have this big long strand of amino acids. Well, how do you know which one goes with which? Well, there's a chart for that. It's called a codon chart. You do not need to memorize this. That would be ridiculous. Somebody probably has memorized. I mean, I'm sure they do. I do want you to know one thing about the codon chart. Well, a couple of things. How to use it for one. And number two, I'm going to ask you to memorize one of the 64 codons. A, U, not T. I just noticed this. And G. There should be U's here, but it's okay. AUG, which is methionine, which is a start codon. Right? AUG is the start codon, and every time you see it, it's going to code for a met or methionine, but it's always the first codon of a gene. Every time it's called the start codon. So if there are start codons, what else is there? Well, there's stop codons also. Let's find them. Here's a couple down here, and there's another one right next to it. There's 3 stop codons, and they don't code for an amino acid. 61 code for an amino acid, 3 code for a stop codon, and what do you suppose the stop codon means? One guess. It means stop. This is the end of the gene. When you get to the stop codon, we're going to stop. Now, what is bringing those amino acids? We already talked about this, tRNA. Let's talk a little bit more about it. Here's tRNA. So tRNA looks like this. There's so many different pictures of tRNA, but this is okay. Look at it. It looks kind of big and proud. Okay, so ACG codes for this codes for a particular amino acid. We could probably come over here and find it. ACG, threonine. Shocking that there's threonine right up here, right? All this stuff is standard. Down here at the bottom, notice you have the complement of this codon down here, UGC. This is called the anticodon, and it's a part of the tRNA that kind of complements that. And so this is going to be specific. This tRNA doesn't go get any amino acid. It always goes and gets this amino acid every single time. So there are actually 61 different tRNAs. They're all just waiting for their number to come up, and they're going to go get their amino acid, bring it in, add it to the pile in here as we make this protein. And so tRNA, that's what it does. Now, if there's a stop, obviously it's not going to bring an amino acid because there's not an amino acid called stop. And so here's some basic steps of this. We're going to just, really, really basic. All right, and so this is where it starts right here. We're just going to pick a place. And so the tRNA comes in. These little blobs are supposed to represent like the amino acid at the top. Here comes another tRNA. It's bringing in another blob, and those blobs are going to connect to that one, and then you have 3, right? And then the other one's going to go away, and now you have this chain, and you can imagine this thing continuing on until you have just this long chain of amino acids. Some proteins can be thousands of amino acids in links. Some not very many, depending on what the protein's function and shape is going to be. But this is going to continue until it gets to a stop codon, at which point that protein is going to break off, and that protein is going to then fold up and make a shape, and that shape is going to determine its function.